Okay, the next speaker is Leo Chouliaras,
Yeah.
and is from the Department of Psychiatry. And the title of the talk is DNA methylation sequencing in Lewy Body Dementia.
Yeah. Hello, everyone. Good afternoon. I hope you can hear me well. So my name is Leo, and I'm based at the Department of Psychiatry. I'm doing 3 days a week clinical work, and the rest of I'm doing research. I'm an old age psychiatrist by background, but today I want to talk to you a little bit about dementia with Lewy bodies and Lewy body dementia and the work we are doing. We haven't talked much about it today, but I think it's a good opportunity to introduce it. This doesn't seem to change the slide. I don't. Oh, now. So what is Lewy body dementia? Obviously, the name comes from the pathology that we see in these patients' brains. I think it was discovered by Professor Spilantini that alpha-synuclein aggregates in Lewy bodies in these people who get Parkinson's disease, if it's mainly in the substantia nigra, but there is also the dementia syndrome of what we call Lewy body dementia. It's essentially two diseases grouped under an umbrella term. We have Parkinson's disease dementia that we heard earlier about it. People develop Parkinson's disease, and then at least a year later, they develop cognitive problems and a dementia syndrome. But there is also dementia with Lewy bodies where people present to the memory clinics or to psychiatrists with a group of symptoms and end up having this disease that essentially, when they have a post-mortem examination, their cortex is full of neurons that have Lewy bodies. Having said that, I'll come back later to that. And the typical syndrome is that people may present with Parkinsonism. You know, all the typical symptom of bradykinesia, rigidity, slowness, and the characteristic walking. Sometimes we see them in the clinic walking in, and we know that could be something like that. But they also have more distressing symptoms. They can get recurrent visual hallucinations. Those people often see animals in the room, they see people, and that can be very disturbing. And the problem with that is if we don't diagnose that the hallucinations are because of that, they may be prescribed an antipsychotic medication and then have very, very severe adverse reactions. Those people also often have REM sleep behavior disorder. Those, I think, with the next one are actually the core features of the disease. What we talk about REM sleep behavior disorder, people act out their dreams very vividly. They often fight in their sleep. They fall out of bed. So it's a very severe dementia syndrome. And people also may present with what we call fluctuating cognition. This can be difficult sometimes in the clinic to get good history about. Often they are very sleepy during the day. They might sleep for more than an hour or two during the day, and they can have periods during the day that are very, very confused. When we do MRI scans, those people may have slightly different appearances on the MRI. Here you see someone with dementia with Lewy bodies who has relatively preserved hippocampus or mediotemporal lobe, while someone with Alzheimer's disease often has mediotemporal lobe atrophy. And this is a suggestive feature, but approximately 50% of people with dementia with Lewy bodies may have coexisting Alzheimer's disease pathology, and that actually makes things more complicated. And sometimes people with Lewy body dementia have differences in their cognitive tests. They more often have visual spatial problems in their cognitive profile, while people with Alzheimer's disease may have more impaired recall. Despite those sort of characteristic clinical syndrome, it's very difficult to diagnose. We know when it comes to how common it is, there is a little bit of discrepancy there. When we looked at brain bank cohorts, autopsy collections, Lewy body dementia can account for approximately 15 to 20% of people who are in brain banks who have donated their tissue. So we think it might be the second most common type of neurodegenerative dementia in older people, accounting up to 15 to 20% of cases of people with dementia. And that's in several different brain bank cohorts. It's something that is widely seen. But when actually we see those people in the clinic, and that was a study in the region here comparing Newcastle with Cambridgeshire, only about 4.5% of the people who came to the memory clinic were diagnosed with dementia with Lewy bodies, while we think we might be missing a significant proportion of them, and that's across different cohorts. So clearly we need better biomarkers for this disease. We need to be able to diagnose it early, especially with the advent of new treatments for Alzheimer's disease. If someone has coexisting Alzheimer's disease and synuclein pathology in the brain, they are very unlikely to benefit from the anti-amyloid treatments, or at least we have to check that. And we really need to have better treatments, better biomarkers for other diseases other than Alzheimer's disease. We study epigenetics. We try to sort of think of what may be causing dementia with Lewy body dementia or neurodegenerative disease. And in general, we think that the model of environmental factors might be affecting that, might be implicated. There is some epidemiological evidence, but also genetics, and it's ultimately the interactions between genetic factors and environmental factors that might be leading to the underlying neurodegenerative process. There are lots of different ways to study epigenetics, different levels. Essentially, we might have modifications on the DNA, we might have modifications on the histones. All of those do affect gene expression and essentially cell behavior and protein changes, but there are also obviously several layers that need to be studied to better understand this. Our focus is mainly on DNA modifications and DNA methylation per se. I'll come back a little bit later as to how we study DNA methylation, but essentially there are some enzymes, there are some enzymes, DNMTs, DNA methyltransferases, that adds methyl groups to the cytosines in the DNA and affect how open or closed the gene is at that point through a complex system, essentially predicting how genes are expressed or how active is a gene or not. And there are ways to study that. And I just wanted to highlight here, in Alzheimer's disease, studies of epigenetics are quite established. They are now at the level of meta-analytic studies with thousands of brains being included and having epigenome-wide association studies following genome-wide association studies. But we are quite behind that when it comes to Lewy body dementia, which is not as rare as we initially thought. A few years ago, we wrote a review summarizing what was known back then. There is some evidence about genetics, and some new things have come through. There is evidence of only a handful of genes having both genetic causes and epigenetic changes, and even less about what happens at the epigenetic levels, clearly showing we need to do more research to understand that. Actually, since we published that, there was only one study that came out showing some more hits that might have differential methylation. So what are research questions? What do we try to do in Cambridge, and what we've done the last few years? One of the things we wanted to look, we do mainly clinical research studies. We recruit people through our memory clinics, and we wanted to know whether we can pick up any of those epigenetic changes in the blood, whether we can use blood as a proxy for what happens in the brain. And then we do that in people who we have seen in our clinics who have very extensive imaging and other data. And blood obviously is very easily accessible, can help us develop a biomarker. But we also know that while it's not the brain, it might be different from what is happening in the brain, that actually there are lots of immune system changes, both in Lewy body dementia and other dementia disease, that we might be able to pick up something in the blood, a signal. But obviously we also want to look what happens in the brain, what really, are there any epigenetic changes in brain tissues, and in particular post-mortem brain tissues that we can access. And ultimately the idea is whether we can combine multiple layers of data when we see people to gather lots of information on genetics, epigenetics, neuroimaging, and immune system changes, and see whether we can understand better this disease or develop better biomarkers. So in this study that we did, we put together blood samples from four cohorts across Cambridge and Newcastle. We had blood samples, DNA from those people, and most of those people had their genetics done. They had plasma biomarkers, which we've published separately, and PET scans as well. And they were, we tried to balance them as best as possible. We had 156 people. And we did DNA methylation sequencing using a method called reduced representation bisulfite sequencing. Essentially, the way it works, how we can most easily measure DNA methylation, is with this method called bisulfite sequencing. We add a chemical called sodium bisulfite to the isolated DNA. And what happens essentially is all the cytosines that have a methyl group remain cytosines after we add this bisulfite, sodium bisulfite. But the unmethylated cytosines, they become uracil. So then if we get this converted DNA and then sequence it in a next generation sequencing machine or using other ways, we can get a readout of what cytosines are methylated and which ones are not. And for this particular method, we used RRBS, as it's called, that essentially using enzymatic digestion, we only pick up specific fragments of the DNA that are dense in CPG regions and might be more likely to show methylation changes as a way of avoiding whole genome sequencing and making that quicker and cheaper. We used commercial kits, and actually being in Cambridge means we are very lucky to have access to core facilities. This work, for example, was done at the SMCL Academic Department of Genetics, where we collaborated and they did the sequencing for us. We ended up with approximately 2 million CPGs that we measured across cases and controls. And to improve our chances of finding difference and have adequate statistical powers, we can actually do region analysis comparing promoters, CPG island source, and CPG islands next to single CPGs that were present in all of our samples, because it's more likely that methylation changes across a region may have more biological effects than in a single CPG base. And we use sort of standard QC processing and methods to analyze the data. And actually, again, coming back to the Cambridge Neuroscience community, a lot of the bioinformatic tools to analyze this data are developed by the Abraham bioinformatics teams who make their software publicly available. And obviously, our study is possibly underpowered. We used FTR to correct for multiple testing. So when we looked at the data using a single CPGs, you see here a volcano plot. The sort of more to the right side is the bigger methylation differences, and to the left, hypomethylated, hypermethylated. And then we obviously set up the multiple testing correction p-value. We had two hits that were significantly hypomethylated, in this case in cases and controls, the EMC6 gene and TNFR-S1FP. They are not known genes to be involved in this process, but no one has looked at this type of data before. The one is involved in mediating effects of TNF alpha, so some kind of immune involvement, and the other one is involved in endoplasmic reticulum membrane protein complex and the autophagosome assembly, again, pathways that we know are important in neurodegenerative diseases. And then when we looked at a sort of regional analysis, I plot here the CPG island source. Again, we had some hits that were significantly hypomethylated. We had ABCG5, again involved in lipid transport across the membrane, ETA4 related to DNA damage, and tenurin4, which again has some previous connections, genetic studies with Parkinson's disease and might be and essential tremor. So we saw that in people that we've seen in our clinic with the diagnosis of dementia with Lewy bodies, we can find DNA methylation changes in their blood that seem to be significant in our small cohort, but obviously need further replication. We looked further into those specific hits. The changes for the two genes are fairly significant. They were in the range of 40% in the controls to 20% in the whole region in DLB in this one, and a little bit less on that. 10M4 was not as marked, the changes. And then we wanted to see, obviously, whether those can be used as biomarkers, whether we can combine them and see if they can differentiate cases from controls. And they seem to be quite accurate when we make a panel of them and do the raw curve statistics. They give us an accuracy of a 0.091 of an area under the curve with a good sensitivity and specificity. But that's obviously only in our single cohort, and we need to replicate that in a larger cohort. And then we also looked whether they can tell us anything about PET changes, PET amyloid changes in this case. We are always interested to know whether people with Lewy body dementia have coexisting amyloid pathology in the brain. So we have PET data with PET amyloid for some of those people. And again, we found that one of those genes have some correlation with PET amyloid status in that respect. And again, there was an area under the curve of 0.84. That's what happens in the blood. I think we have some preliminary evidence that we can use DNA methylation sequencing, perhaps with some potential as a biomarker. And then we obviously are in the process of getting funding to replicate it in a larger cohort to be able to see the significance of that. But it's even more important if we want to understand the disease better to look for epigenetic changes in the brain. And then while we were doing the work in the blood, we also had some access to post-mortem brain tissues. We actually, again, were very lucky to get some tissues from the Cambridge Brain Bank. That was the initial to establish our protocol and test everything. And then we got a cohort of people with post-mortem brain tissues from the Newcastle Brain Bank, and we got 60 samples with dementia with Lewy bodies and controls. We've got frozen sections from the cingulate gyrus because that's one of the most affected regions in the disease in terms of alpha-synuclein pathology. And we used alpha-synuclein immunohistochemistry. So in frozen tissues, we labeled neurons with Lewy bodies and neurons without Lewy bodies using a Nissl counter stain and the alpha-synuclein antibodies. And then we used the method, again, something that is available in Cambridge at the core facilities, a laser capture microdissection microscope. And then in tissues, in the brains, we could get neurons that have Lewy bodies and neurons that were free of pathology, cut the DNA out, and sequenced DNA methylation to look for epigenetic differences. And we had two comparisons, neurons from control cases and neurons without pathology from cases with dementia with Lewy bodies to see if the neuronal DNA methylation changes in cases versus controls, but also from the same people, neurons with pathology and neurons without pathology. I have a video here with how the laser microdissection microscope works. I'm not sure if that will work because it didn't work earlier, but I'll walk you through it. Essentially, this is a frozen section of post-mortem brain cingulate gyrus, and then you can see here a neuron with a Nissl stain. That is a neuron that doesn't seem to have any pathology, while you see here two neurons that seem to have alpha-synuclein pathology. And what you can do with a microscope, you can cut out this neuron and it falls in one tube, and then you can cut out those neurons and fall in another tube. And what we did that we figured out from our pilot testing, we needed about 1,000 neurons for each sample, so more or less a day's work to produce enough DNA to do our comparisons. Yeah, the video doesn't seem to work, but essentially what you would see is the laser cutting through here, and then this falling behind, and then those falling in another tissue. So we ended up with samples that we had neurons purified with Lewy bodies and neurons without Lewy bodies, and then compared the DNA methylation profiles. Again, we used bisulfite sequencing. In this case, we couldn't get the reduced representation sequencing to work, so we had to use a modified protocol for whole genome bisulfite sequencing, so slightly different analysis compared to our blood samples. Bigger sort of more DNA, more DNA sequence, but with the damage of those tissues, we couldn't get accurate sort of whole genomes. And again, we did regional analysis and single CPG analysis to compare cases with controls. I've put here some preliminary findings from the case control comparison. I'm still processing those and need to establish exactly what is more relevant and what is found in other studies. But again, we seem to find more established changes in the brain, obviously, because it's the tissue of interest and there is less variation in terms of cell type populations. But there are, again, some genes that seem to become hypomethylated or hypermethylated associated with the presence of Lewy body pathology. So in summary, I hope I gave you a snapshot of our work, showing that we can detect that Lewy body dementia is associated with DNA methylation changes in blood and brain tissues from patients with the disease, and that we can use DNA methylation sequencing to find new genes that are and pathways that are associated with the disease and help us to understand the disease better and develop new ways of diagnosing and treating it. And obviously, with this kind of work, we always need to replicate it in larger cohorts. And we are collaborating with the University of Exeter and other cohorts to establish that. Obviously, I'd like to thank all of our donors and people who have given up their time and their samples, and all of our collaborators and the funders for this work.